Okay, so in the notes, remember this section, or this chapter I should say, is very, very short. There's only 2 sections. And so just in the 40-minute video from last night, we're over halfway through with this chapter. It's a very quick chapter. And so if you didn't watch yesterday's video, you need to do that, all right, because you've missed more than half of the chapter, okay? So, so the first part was, as we talked about a couple of days ago, that some experiments proving that DNA was the genetic material. And then we learned and reviewed about the structure of DNA. And then now we're in the second section, which is about how DNA replicates. So how do we go from, remember why DNA needs to be replicated is, for instance, when we divide by mitosis, our cells have 46 chromosomes, therefore 46 pieces of DNA. When it divides and we grow, each cell needs to have 46 pieces of DNA, and so therefore we have to replicate the DNA, and this is the process by which that DNA replication happens, all right? So that same process occurs, DNA replication in meiosis as well, and so that's the purpose of DNA replication. So when DNA replicates, remember DNA is a double helix, so it's a helical molecule. So before this actually gets here, to this picture here, the DNA has to unwind, untwist, and then the 2 sides of the DNA have to break and come apart. The original sides here are the darker blue here. Those are the originals. They have to, sometimes they call it unzipping it. What kind of bonds are broken when that happens? Hydrogen bonds, correct? And so, so we'll learn in just a little bit here that there's also some enzymes that help with that untwisting and breaking of those hydrogen bonds. And then once we have the 2 DNA strands separated, we add nucleotides. So coming out here, sticking out here, this right here is a chain of nucleotides, which is a phosphate, sugar, and a base. So if you have an A here, that means that to build the second strand here so that we replicate it, that nucleotide would have to have a T. A's go with T's. If this was a C, you'd have to have a G, and so on and so forth. And so the enzyme that helps with that is DNA polymerase here. DNA polymerase 3 adds nucleotides and grows the nucleotide strand, and so we have 2 sides that we need to replicate so that we end up with 2 identical pieces of DNA from 1 piece of DNA. So now we say that this one's the leading and this one's the lagging strand. Can somebody tell me what the difference between those are? The leading and the lagging strands. Megan?
The leading strand is going in the direction of towards like a Y-shaped thing, and then the lagging strand is the nucleotides are added in fragments because they have to be going in opposite direction of like the bubble, and they have to like go back and then make the nucleotide bases in fragments.
That's correct. So she was talking about this Y-shaped here. That's called the replication fork. And so the leading strand moves, it adds nucleotides in the direction towards that replication fork. The lagging strand is in the direction opposite. Now let's talk about why that is. It has to do with some kind of pickiness with DNA polymerase 3 when it adds nucleotides. DNA polymerase 3 can only add nucleotides to the 3' end of an existing DNA molecule. So therefore, when we look at the original DNA, so this is the original, this is the 3' end, this is the 5' end. If we look at the end here, so 3', this is the 5', this is the 3'. So therefore, they're running in opposite directions. That's called antiparallel. And so that's your original DNA. So therefore, when we replicate this DNA, if this is this side, that means the end over here has to be the 5', and if this is the 5', the end here has to be the 3'. So that we know, so that the 2 new strands that you're going to build are antiparallel as well. So what happens is this DNA polymerase can only add nucleotides to the 3' end. So if this is the 5' end, this must be the 3' end of the new strand here being built. And therefore, DNA polymerase can only add nucleotides to that 3' end. So therefore, the new light blue strand is built in the 5' to 3' direction, adding only to the 3' end. Now, so that doesn't pose a problem with the leading strand because this is the 5' end, this is the 3' end, and so it keeps on adding nucleotides. It moves towards the replication fork and just continuously makes that DNA strand. When we look at the lagging strand here, we notice here the original strand, this is the 5' end. This needs to be then the 3' end. And remember, the new strand has to grow in the 5' to 3' direction because DNA polymerase can only add to the 3' end. And so therefore, what you have to do is the DNA polymerase has to start here, this is the 5' end, and add nucleotides in this direction to the 3' end until it gets to the end, and then it has to start over again here and build this one in the 5' to 3' direction, and so on and so forth. And that's called the lagging strand, and the lagging strand, those fragments are called what? Okazaki fragments, based upon the Japanese scientists who discovered them. All right, so you get these fragments, and then down here you notice that the leading strand, it unzips some more of the original DNA. The leading strand just keeps on plugging away, adding to the 3' end, whereas your lagging strand, every time the enzyme gets to the end, it has to come over here and start again, and you get these fragments. Well, we don't want them to actually stay as fragments, so in comes another enzyme called DNA ligase that actually bonds them together, so it hooks these guys together and actually kind of quote-unquote glues them. It's kind of a little analogy there. So then eventually you get one continuous copied strand, and therefore you end up with 2 identical pieces of DNA molecules. So last night in the video, I talked about this, but the bullets previously, there are 3 bullets previous to these that I skipped. And so what I'd like you to do is go back to those 3 bullets and now fill them in. So if we go back, these 2 bullets I had given you, that DNA is antiparallel, and that means that the sides, one goes from the 5' to 3' direction, and the other one goes from the 3' to the 5' direction, so the sides are opposite. And we just talked about how DNA adds nucleotides only to the 3' end, and therefore the new strand grows from the 5' to 3' direction. So then the next bullet is what you need to fill in. So along one template strand of DNA called the leading strand, DNA polymerase synthesizes a complementary strand continuously. So leading strand, the key word there is it's synthesized continuously, doesn't have to stop, moving toward the replication fork. And then to elongate the other strand called the lagging strand, DNA polymerase works away from the replication fork. And because of that, and because the DNA polymerase can only add to the 3' end, the lagging strand is synthesized as a series of segments called, and you should have Okazaki fragments, which are joined together by, and that is DNA ligase. Okay, so how'd you do on that? Okay, so we're going to go ahead and move on here. So with that same picture, we're going to add a little bit onto it that it didn't add on, doesn't show on that picture. So this is called priming DNA synthesis. So DNA polymerases cannot initiate the synthesis of a polynucleotide. They can only add nucleotides to the 3' end. So they can't initiate it. So they can't start. A polynucleotide, poly means many, so it's many nucleotides. So one side of a double-stranded piece of DNA, one side is a polynucleotide, the other is a polynucleotide, the hydrogen bond in the center. So what this means, going back to this picture, is if we look at the leading strand, the way this is drawn, it makes it seem like this DNA polymerase at the very beginning, before all this was built, this DNA polymerase can bind to the end and start adding nucleotides and be all good. That actually is not true. DNA polymerase can't initiate the synthesis of a polynucleotide. So it can't be the first one to add a nucleotide. And so therefore, that's a problem because we need to have it start somewhere. And so, so how do we solve that in our cells? The initial nucleotide strand is a short one called an RNA or DNA primer. In us, it's an RNA primer. Let's do a quick little, we've talked about DNA and RNA before. So it's a nucleotide strand. I'll write it over here. What is the difference between a nucleotide in DNA and RNA? Do you guys remember the 3 parts to a nucleotide? All nucleotides have a phosphate, a sugar, and what sticks out? A base. All right, so those are the 3 parts to a nucleotide. RNA and DNA are both polynucleotides. They're chains of these nucleotides. What differs between RNA and DNA nucleotides? Do we remember that? One thing is the sugar. Lucas?
The sugar is ribose, and the difference between the bases is that instead of a T, there's a U or something.
Yeah, ribose versus deoxyribose. Ribose is the R in RNA. Deoxyribose is the D in DNA. All right, so whether or not which sugar is there. And then the bases, in DNA, you have A's, T's, C's, and G's. In RNA, that T, remember, is replaced by a U. Okay? So those are some differences there. So what happens is the initial nucleotide strand is a short one called an RNA or DNA primer. So like I said, for us it's RNA. So therefore the nucleotides are just a little bit different. And what happens is this enzyme, another enzyme called primase, can start an RNA chain from scratch, unlike DNA polymerase. So that's why it's used. Can start an RNA chain from scratch. So let's go back. I'm going to use my red color here. And let's go back to that picture, and we're going to add onto it what they did not put onto this picture here. So if we look at here, here we have the leading strand. So we just said that DNA polymerase can't add and be the first to start adding nucleotides. So what happens is this enzyme called primase can start the nucleotide chain. And so primase adds an RNA primer here. So I'm going to go ahead and just color this end red to show that this is not your DNA nucleotide. So let's write here that this is a primer. In us, it's let's put an RNA primer. RNA primer added by, what was the enzyme? Primase. And so then, since primase can add and start a polynucleotide chain, then we're good to go, because now you've got a little short, it's very short, a little chain of nucleotides, and now the DNA polymerase can add, because this is the 3' end of this little strand here, start adding, and then it continues. And so therefore, for the leading strand, you just need 1 primer at that very beginning, and then the DNA polymerase is good to go. Let's look at the lagging strand. The lagging strand, remember, were built in fragments. And so, so if we look at this fragment here, where would the primer be? Would it be at this end, like it is here? Where would the primer have been added? Yeah, to the very beginning here. So therefore, so when the DNA unzips here, the primase can add a primer here, and DNA polymerase can add it continuously. But here, you have to have primase add a little primer here. And then DNA polymerase can add to the end. And then it unzips some more, and that's not a problem for this. The DNA polymerase can continue to move continuously there. But here, what do we need another of? We need another primer. At the beginning of every Okazaki fragment, you need a new primer. So therefore, it's kind of hard to see it in here, but I'll just, so you have a little primer in here. And then it needs a new primer, and then DNA polymerase can add the nucleotides till it gets to the next Okazaki fragment, then it unzips some more, you have to have a new primer, and then DNA polymerase adds, and so on and so forth. So let's label these guys as, these guys are primers too. So every time, for every fragment, you have a new primer. So that's new onto this picture. So then if we go back to the last bullet here, well, actually, you should be able to fill in that. Go ahead and fill in that last bullet. Each Okazaki fragment must be primed separately. So you have many primers there. Question?
Is the chapter 16 review going to be due on Monday or Tuesday?
No, I just want you to start working on it.
Okay. When's the test for that?
Oh, it's not next week, but because we're combining, we have to do all 17 too.
Okay.
Yeah.
So like the next week.
Yeah, so probably the following.
When you're gone, probably.
Okay. So let's look at this picture here. This picture kind of just illustrates what I kind of drew in the other picture. I like the other picture because it shows both sides of the DNA. When it opens up, this is just showing you one side of the DNA. So, so here is the 3' and the 5' end. So therefore, here is the primase making the primer. So it's dark on your sheet here, it's red on mine. This is then, since this is the 3' end of this original DNA, this has to be the 5' end of the new, and this is the 3' end. So therefore, the DNA is going to replicate and go in this direction because remember, DNA polymerase is going to add nucleotides to the 3' end. So here's your primase adding the primer. And now DNA polymerase, and then the next step is DNA polymerase now can add nucleotides to the 3' end, because this is the 5' end, so it's adding it on there until it gets to the end, and then it comes off. So this is your first Okazaki fragment, if this is the lagging strand. And then remember that it has to go back and build another Okazaki fragment. And so what this is not showing you here, it says it in the reading, what do I do with my red marker? It says in the reading up above here, it says after the second fragment is primed. It just doesn't show the primer, so I'm going to go ahead and just show that here. So here's your primer, and then DNA polymerase can add nucleotides there. And that's your second fragment. Then what we haven't talked about yet is what do we do with this RNA nucleotides? RNA nucleotides are different than DNA nucleotides. So therefore, there's a second DNA polymerase. The DNA polymerase 3 added nucleotides to grow your strand. DNA polymerase 1, what it does is replaces or takes out the RNA nucleotides and plugs in DNA nucleotides. So that's what it's doing there, and that's why this little primer is now shorter, because it's replacing the RNA nucleotides. And then last but not least, which we have talked about, is then DNA ligase comes in to quote-unquote glue the strands together, so you have all the Okazaki fragments, one continuous piece of DNA. All right, and so that is another way, another picture to look at that same process that we just looked at. All right, adding in this new DNA polymerase 1. All right, so then, so that's replication in a nutshell. There are a few other enzymes that, or other proteins, which enzymes are proteins, that help with DNA replication. So I alluded to the fact earlier before DNA replicates that it has to unwind and unzip. So there's an enzyme called helicase that does that. It untwists, so that's what I was talking about when you untwist it and the hydrogen bonds are broken. Helicase helps with that. The next thing that's new is we have these things called single-strand binding proteins. Now there are different ones of those, but we just need to know that name. Binds to and stabilizes the single-stranded DNA until it can be used as a template. What does that mean? When you untwist and break the hydrogen bonds. You don't want them to go back together, so these single-stranded bonding proteins kind of stabilize it so that the two sides don't re-hydrogen bond together. They stay apart. That's what those do. All right. What time do we get out of here? 27? All right. You guys want to stop here? You just watch the rest of it? Yeah. All right.